Good morning, everybody. Thank you for inviting and thank you for being here. I'm very excited to be here again. I was here 11 years ago, and obviously there was no campus yet, so I'm really thrilled to actually finally see it. As Chris said, my lab is interested in the response to proteostatic stress. And before I dive into that, I would like to define what is actually proteostasis. It's nothing but like the balance of a healthy proteome in the cell. That could be the abundance, the interactions, the functions, the folding, and everything else about the proteins. We just had a wonderful talk about the DNA, so this is the other end of the spectrum. And very simplistically speaking, proteostasis is regulated by the synthesis and the degradation of the proteins. Obviously, there's more processes. And then it is tightly linked to everything else in the cell, so also to cellular homeostasis, meaning how does the cell do, right? The cellular health. And if the proteome is imbalanced, then that is detrimental for the proteins themselves. They accumulate, misfolded, unfolded, damaged, or misexpressed proteins accumulate, and then trigger what is known as partially as the ER stress response, or also as the unfolded protein response, or the UPR. And yesterday, we actually already had a couple of talks mentioning the UPR, and I will allude to other aspects of it. And then unsurprisingly, if you have proteostatic stress, imbalanced proteome, cellular homeostasis is compromised, and this is linked to a number of diseases, cancer, inflammation. And in our case, we're particularly interested in the link, in the strong link between proteostasis control and neurodegeneration, meaning a very fundamental question in that area is like, why are some neurons particularly sensitive to proteostatic stress and others aren't? Do I have a pointer somewhere? Ah, okay. All right. Okay, so throughout my talk, you will see sometimes like the blue or teal neurons that are degenerating, and then I read the kind of similar but surviving neurons. And so ER stress is, because of its central role, regulated in three main pathways in the cell. And this is why, to a systems biologist like me, this is highly interesting because it's complicated. Right? So it all starts with an accumulation of misfolded proteins in the ER. Obviously, it's never as simple as that, but for the sake of this talk, let's just say that. And so one thing the cell tries to do is ubiquinate and degrade those erroneous proteins in a process called ERAD, or ER-associated degradation. Obviously, there's also a removal of the erroneous RNAs going on, so RNA degradation. And then there are these three main pathways. One involves a nuclease, IRE1, that then splices XPP1, which is a transcription factor that goes into the nucleus and triggers a transcription response. The second one, Michael alluded to yesterday, is governed by ATF6, which is also a transcription factor that after processing in the Golgi goes to the ER to trigger transcription response. And the third pathway, centered around PERK, a kinase, is what we're particularly interested in. It involves a phosphorylation event that first phosphorylates PERK itself, and then EIF2 alpha. EIF2 alpha is symbolized by this little purple egg here. It is a component of the ternary complex, which itself is a component of the pre-initiation complex that is essential to start translation in the cell. Right? So initiation factors in the translation work are abbreviated as IF, and then EIF is a eukaryotic translation factor. And so phosphorylation of EIF2 alpha by PERK lowers the abundance of unphosphorylated EIF2 alpha, which then leads to this global shutdown in translation. And again, Michael mentioned that yesterday because he had this surprising finding that translation was actually upregulated in his system. And even in the normal ER stress response, there's actually exceptions to the theme, and I'll get to that in a little bit. There are genes that are actually translationally induced in response to ER stress, despite the global shutdown of translation. So by origin, as Chris said, I'm a proteomics person, I'm a systems biologist, so we wanted to study this highly complex process in its dynamics. And so you set up an experiment where you take your cells and you stress them and you look at different time points. And we collected the RNA data. We looked at ribosome footprint data, so meaning like the footprints of the ribosomes associated with the mRNAs as a readout for translation efficiency. Of course, we collected the protein data. And when you do that, you have two things that are a little bit more complicated than what you normally do when you do gene expression analysis. One is you have to deal with time. So when you normally look for differentially expressed genes, you could say, OK, I have a certain fold change or a significant p-value. But something that's very, very small might not be significant on its own. But if it's consistent over time, you still might consider it being significant. So you need to incorporate that time component. And the second thing you have to do is, if you measure these different types of omics things, you have to incorporate the relationships between these different data sets, right? Obviously, the number of proteins that you observe at any given time is a strict function of how many mRNAs you have present, right? And similarly, the number of ribosomes bound to an mRNA is a function of how many mRNAs you actually have. So already a few years ago, when we decided to venture into this, we realized we had to actually develop the tools to analyze this data. And to cut a long story short, we did using very simple rate equations that model this synthesis and degradation of these molecules in their dependencies on each other. So for example, for the protein case here, you would model it as a function of translation based on the mRNA that's present minus the degradation based on of the protein that's already present, right? So you can incorporate time and the relationships between the molecules. And so already many, many, many years ago, we did this study, but that was really central to everything we're doing in the lab, where we did exactly this. We collected mRNA data in HeLa cells stressed over 8 hours with an ER stress-inducing agent. We collected the RNA data, the ribosome footprint data, the protein data, and because it wasn't crazy enough yet, we also looked at the data that describes the footprints of the non-ribosomal proteins bound to the mRNA. And because this has been published a while ago, I will only show you one figure from this, which is the main heat map, to illustrate the complexity of the response and I think, the beauty of these data sets. This is what we looked at and analyzed here. We have these 4 different omics data sets. We have like little time series experiments. We have them measured them in replicate. Red is up, blue is down. We normalized everything. And what you can already see is that there is, for example, a good transcription response. Sometimes it's measured, it's matched by a translation response, sometimes not. You see changes in the protein mRNA occupancy corresponding or not corresponding to that, as well as the protein level. What I do want to tell you about today is 2 extensions of this work that we're currently still actively looking at. One took this system into neuronal cells. And this is where we teamed up with Esteban Mazzoni, who is also at NYU Biology. And he's able to differentiate stem cells into lots of cool motor neuron types. And this is relevant to a specific neurodegenerative disease, specifically amyotrophic lateral sclerosis, or ALS. One of the most famous patients who passed away from ALS is Stephen Hawking, and Lou Gehrig, who gave the disease its American name, also obviously was a victim to it. It is a fatal disease. It affects 1 to 3 people per 1,000, a population of 1,000. There's no single gene cause. 95% of the disease are sporadic, only 5% familial. And for the familial cases, we only have a very limited understanding of what causes the degeneration, mostly of the spinal motor neurons. The spinal motor neurons are the motor neurons that regulate, like attach to your muscles and enable you to move. And so this is why the patients get paralyzed. However, a big question in the field is, why can patients, including Stephen Harkin, actually move their eyes until late stages of the disease? So in other words, why does a subset of these cranial motor neurons, which in principle are very similar because they're also motor neurons, survive until late stages of this disease? And this is really central to a lot of things that Esteban looks at and that we're looking at in our lab from the perspective of the ER stress response, because ER stress is highly linked to neurodegenerative diseases. And so Esteban was able to differentiate embryonic stem cell, mouse embryonic stem cells, and now human-induced pluripotent stem cells into these cranial and spinal motor neurons and conduct these controlled experiments and amazingly show that indeed cranial motor neurons are actually, these induced cranial motor neurons are actually also more stress resistant compared to the spinal motor neurons that are more sensitive to the disease as well, right? So it's a similar color scheme as I showed you before. Here the red cranial motor neurons have a better survival in response to CPA-induced proteotoxic stress compared to this induced spinal motor neurons. And so this is where we teamed up and we said we now have a perfect system where for the first time we can actually do these controlled studies and look at the differential stress response in these two cell types. So again, we conducted a time series experiments. We only collected RNA and protein data, but this time for the two cell types in parallel. And so the data set that I'll show you in a second now has this additional dimension where you see one cell type on the left, the spinal motor neurons, and then one cell type on the right, the cranial motor neurons. You see RNA and protein data. You see no stress in 12 and 36 hours. And again, we conducted the experiment in replicate. We were able to collect complete data for over 8,200 genes, and that's shown here. Again, the analysis of that, actually, it was most of the COVID lockdown that I spent staring at this. The analysis of that is obviously very complex because you can look at this data in several ways. You can look for differences across time in stress, no stress. But most importantly, and that is really what we're interested in, is the differences between the two cell types. And so what you can see is that, well, the two cell types are actually very similar. You have pretty much the same thing on the left as on the right. The second thing you see is that RNA and protein expression patterns are different, which is nice for proteomics people to say, well, it's worth going through the pain of doing the proteomics work. And then once you look more closely, you start seeing some patterns where, for example, here there's an expression change that is not exactly monitored in the other cell types. And this is what we then looked for, where are the differences between the cranial and the spinal motor neurons. And again, I want to cut a very long story short and jump to one of the major insights. One thing that we saw was this expression pattern. So this is like a zoom in to this big matrix, which has exactly the same layout, spinal and cranial motor neurons. But now I'm only showing you the subunits, the proteins that code for the barrel, the core of the proteasome, and then the regulatory subunit, which is basically the lid of that big trash can, right? And you don't need statistics, even though we obviously applied some statistics to this, to see that the expression pattern of the core and the regulatory subunit are really, really different. And this puzzled me for the longest time. Because what this means is there's some sort of weird relationship between the abundance of the fully assembled proteasome, which is able to recognize these ubiquitinated proteins, these damaged proteins, right, and degrade them. And then the actual core proteasome, which actually can also degrade proteins, just that it's incapable of recognizing ubiquitination, so it just degrades whatever comes along its way. So a lot of kind of forth and back and quite challenging experimentation allowed us to demonstrate that indeed cranial motor neurons have more 26S proteasome and more active core proteasome. But even more excitingly, we did find a protein in our data set that differentially regulates the activity of the 26S proteasome in the two cell types. And the reason this is interesting is because obviously our system, while still cells in a dish, is linked to this overall question related to disease, which is why do cranial motor neurons succumb early? Why do cranial motor neurons actually show more resistance to ALS compared to spinal motor neurons? So any gene that marks the difference between the cell types is interesting. And so this gene that we identified is called UBL-CP1. It is shown here in purple, and then above this is shown the stressor, no stress, and now we found an inhibitor for UBL-CP1, so we were able to inhibit this protein in the cells. And so UBL-CP1 is one of the few, well, it's the only known nuclear proteasome phosphatase. And what we were able to show is that it differentially affects the 26S in spinal motor neurons compared to the 26S in the cranial motor neurons. And so in the interest of time, I'm just going to cut to the results. In the spinal motor neurons, you actually don't see a difference when you inhibit UBL-CP1. But in the cranial motor neurons, you do see a dependency on UBL-CP1. So the actual work is a bit more complex, but I just wanted to jump to that big finding. We have a gene that marks the difference in proteasomal activity in the two motor neurons. Now, you might be like, OK, proteasome, fine, and nucleus, why is this important? Well, neurons are post-mitotic. They can't renew. They don't divide. And so the nuclear proteostasis is particularly vulnerable to any challenges because they can't renew the proteins. They can't renew the proteasome itself. So they have to really finely regulate the abundance of the proteasome as well as its activity. What I did actually also want to tell you about, and that's the completely different end of the story, is another extension from this project from 2018. And this relates to another aspect of the ER stress response. So in my first overview result, I kind of alluded to protein degradation. But what I want to get back to now is this translation regulation aspect. Whenever you look at the ER stress response and you have ribosome footprint data, you always look for ATF4 because it's a key gene that is known to be induced at the level of translation in response to stress, despite this general shutdown of translation because of EIF2 alpha phosphorylation. And so this is ATF4. It's a prime example of a gene that's induced at the level of translation. What you see here is a picture or a depiction of the main open reading frame. And then this is the UTR, and it has two so-called upstream open reading frames, or ORFs. ORFs are small open reading frames, and the models basically says that under normal conditions, when you have a lot of EIF2 alpha around and a lot of ternary complex, the ribosome starts scanning here, initiates at the ORF here, then reinitiates at the ORF here, and misses this main translation open reading frame because it's out of frame. Right? And so this is why, under normal conditions, you do not have translation of ATF4. And that's good because it's a transcription factor that's supposed to upregulate stress-related genes. And so then under stress conditions, you have low abundance of EIF2 alpha. And so the model that has already been published 30 years ago, and it's really a standard model in the field, and says is because of the low abundance of this ternary complex, you can actually, the small subunit cannot read the AOGs here, and AOG here misses it, but eventually it will have a ternary complex and then initiate translation here. That's in a simplified way the translation induction model for ATF4. So of course, we also looked at this in our stress data. And so what you see here, these are the ribosome footprints. And after 8 hours of stress induction, you indeed see more footprints here. So you have translation induction. And UORF1 here has a decrease in footprints. So this is consistent with the model. But what my student literally did is one day he walked into my office and said, I need to show you something. There's an additional peak upstream of this UORF. And we were very puzzled by this. and I'm running out of time already. This UORF, this additional peak looked like this. It had exactly the width of a ribosome, and in the middle of it was a start codon followed by a stop codon. And this peak here was larger than any other peak in the data, so it was really interesting. And after a lot of forth and back, we found out that it's real. These start-stops are highly conserved, and this is what the remaining few slides will be on. I added the following slide to demonstrate to my colleagues who look at plants that this actually has been observed in other systems. So this short UORF in ATF4 had been seen, but it had never been described in its function. It had been observed in a plant gene involved in the response to boron-induced stress, and had been characterized there as essential for translation upregulation in response to, when you have no boron. And it actually has been seen in HOXA11, a transcription factor involved in development. But nobody really knew what it does in ATF4, and so we set out to characterize this. So one thing you would obviously do is you delete the start-stop here, and when you do that, you find a translation derepression. So this is the mutant start-stop, and you have a translation derepression under normal conditions and under stress. And the important thing here is that the stress inducibility of ATF4, which is crucial for its function, goes down if you do not have the start-stop. And this finding was confirmed by a lot of other constructs and also CRISPR knockouts, which I'm going to skip in the interest of time. I'm going to show you the model that basically expands what had been known about ATF4. So under normal conditions, the ribosome would stall here, and it binds here. And what that means is it represses the useless, not useless, but like the translation of these UORFs. And while the UORFs themselves are important in order to divert the ribosome, you actually don't want to produce all this peptide here because you don't need it for anything, and it's wasteful, right? So you would still have translation repression of the main open reading frame, but you don't make all these U peptides in vain. And then importantly, what we could show is that the start-stop is then crucial for the inducibility under stress, mainly because it represses the second UORF. And so what we then asked is, do we find more of these start-stop elements? We did that using a control where we looked for the sequences with the swapped nucleotide here. And indeed, we found a lot of genes, with ATF4 being the top one, that seem to have this element. In general, they seem to repress translation of genes. And they seem to occur in all sorts of different contexts. So at the top here, you see ATF4 with the two UORFs. At the bottom here, you see a number of genes where there's only a start-stop in the middle. And then you have all sorts of versions here. 50% of the human genes have UORFs predicted. So it's not surprising that there are all sorts of complex constellations. Interestingly, start-stop containing genes are highly enriched in genes that function in various signaling pathways that occur in some SMAD genes, for example, and transcription factors. Okay. All right. We knocked it out in MOR4L1, and we knocked it into ACT-B. So both the gain of function and the loss of function controls work. Okay, because I'm out of time, I'm going to skip the mechanistic aspects that we looked at and jump to briefly to specific genes that we're now looking at. So in addition to understanding how start-stops work, I would like to mention two genes that I got very excited about. One is DRUSHA. I guess everybody has heard of DRUSHA for its role in microRNA function. It's actually also linked to the stress response and, interestingly, to neurodegeneration. So we ask a very simple question. What function do the start-stops have in Russia? But when we looked at it more closely, we realized that its UTR is extremely complex. There's several other ORFs in there. Nevertheless, the second start-stop in Russia is functional. If we knock it out or mutate it, you have a de-repression and translation. And it's also essential for the stress inducibility of the gene. So here you have the wild type induced under stress, and here you do not have this induction if you have the mutated start-stop. And the final gene is, just because it's so cool, I want to mention C9orf72. If you're not from the ALS field, you will have never heard of this, and you will likely forget about it later. It's a guanine nucleotide exchange factor. It activates small GTPases, interacts with 2 other proteins. It has various functions. It's not actually very well understood. Yeah. And it's mutated in 50% of the ALS cases. That is because of a repeat expansion. Okay, I need to stop this before I get thrown off the stage. It does have a start-stop, right there, and I was so excited about it. All right. Okay, we mutated it. It works. It's amazing. There's 2 variants that are clinically relevant. We mutated those. We found start-stops, no, URFs that are functional, so it's all crazy and cool. All right, sorry about this. I told you about way too many projects, mainly about the start-stops that are one regulatory element that affect translation, and it all goes back to our interest in proteostasis regulation and in the context of neurodegenerative diseases. And these are all the other things that we're interested in, with this as the main focus of this talk. Thank you to all the people who did the work, funded the work, and thank you for listening.